family affair.
Yeah. Okay. Maybe later, and let's turn on the camera and okay, so that hi. Oh, it's you again. Okay. You've been here the whole time. Do any of you guys actually know how to make it so that he sees from the camera that's up there?
That's a camera?
Well, isn't there?
That's a microphone.
Okay, that one. A camera. There is a camera in this room, and when people give talks, they can see the professors and the whole class. You know what I mean?
Yeah.
And it moves around sometimes.
Is there maybe another remote up there that it can connect to? Oh, hello. Hello.
Let's see. Okay, well, I'm giving up on that. Hi.
Hello, can you guys hear me?
Hello,
There we go.
I
Okay.
can hear you.
Okay, good. Thank you.
Can you guys hear him?
Yes.
Yeah, good enough.
Yeah, good enough. I think that's the best you're gonna get
It
in
is
this room.
probably the best that I'm going to achieve in this room.
More than most of us.
Well, I'm really glad that you're able to make it work with all the chaos.
Yeah, sorry I can't stay for the whole thing, but I figured that a little bit would be better than nothing.
Yeah,
Yeah,
absolutely.
we just had a little emergency with the kids and we're just trying to figure it out.
I'm sorry. That sounds very stressful.
Kids, they get hurt. You just have to figure it out.
Change the audio source.
I would love to. If you know how to do it, please, please replace me. I'll get out of the way.
Hey, Paolo.
Hello, Candace. How are you?
I'm good.
What a pleasant surprise.
I told them that I know you.
So you should have told them as well that they would be better off without me presenting.
I don't think that's necessarily the case.
How are you doing?
Mm. Yeah, I'm not exactly sure how to get connected to.
Whenever the yeah, usually a lot.
Computer, but I don't know for a Mac specifically.
So I've connected two of the things.
Okay.
One of them got us onto that guy.
Yeah, the projector.
The other one seems to just be juice.
Yeah, that's why I'm not 1000% sure if it has something to do with one of these. Because it might, yeah, and you don't have another, but there's a dongle here. So do you need one to charge your computer?
Uh, maybe, but probably not desperately.
Because I think that's what this would be for. Your Mac is what I think makes it a little bit.
Yeah, oh wow. That's a good sign.
Yeah, maybe. I don't know if you guys know this, but I'm getting a PhD. Sometimes you can just figure things out from playing with it. Now, I'm not sure. You have your Mac is
FaceTime HD camera things.
yeah, I don't know how to
There you go.
Nice.
It's a little angle up a little bit.
yeah, it's angled, which is not great.
Everybody just switch over here.
Yeah, I think there might be a way to move that.
Yeah. Jesse said who's going back to lab after too.
Able to move it. That's the projector.
Yeah.
Pan, tilt, reset, pan, tilt. Yeah, these are all the tilting features, but I don't know how to actually get it activated.
I could stand up and use it.
Yeah, you might be able to, but I would be very careful with making sure you don't accidentally. Yeah, I just don't even know if this properly has, there are batteries in it, but they look pretty old. Sony batteries. What an interesting concept. Probably the OG batteries that came with this.
There you go.
And then you can bring it, are you able to bring it down a little? Okay.
That's pretty good. That's pretty good.
Yeah, so we'll be able to see Dr. Perez and then also hear you very well. Are you able to talk a little bit, Dr. Perez?
Yes, can you guys hear me well?
That's perfect.
Absolutely.
That's so good. That's so much better than before. Okay.
Jesus Christ, that's a big face up there. I'm
Do we want to turn it down just slightly so it doesn't crackle? I don't know how on your computer.
Okay.
I'm a computer person, but like not a Mac person.
Okay, I'm a Mac person and I'm the opposite of the computer person. Okay. Awesome. Okay. How about that?
is it better?
Yeah, I mean, I think it's fine. Yeah? Okay. Okay. Hi, Caroline. I'm not gonna shut it completely, just so that the people who are working in their offices aren't bothered. Okay. Yeah. Okay. No. Awesome. Paolo, could you please try to share your screen and I'll make sure that you have access to do that before I go back down.
Yes. Let me just pull up something here then.
When I was in Physiology, Dr. Oliver, I heard like, set up to a computer. I think Jay was something, but most people don't sound out, so which is fine.
Where is this on? Is it showing there?
It is.
There we go.
Hooray.
I can see it.
Perfect.
There's hot water available. Back here. You're good. Yeah. All right.
I think, are we going to get a level? All right, I think we're ready.
Oh boy, what did I do? All right, so Sophia, just before I do anything, how do you want to do this? Because I was thinking more like making it a conversation
Yeah, I think that's
rather than anything else, rather than, just presenting data and whatnot. How do you prefer?
I think that's really great. I think the big part is that it's nice for us to have exposure to methods and concepts that we don't see in our department all the time. And that's really why I asked you to come here, is because you have this electrophys knowledge and this vasculature knowledge, and it applies to our world, but we just don't
Mhm.
have exposure. So having a conversation around that and learning about that would be really great.
Okay, so let me pull up another one then, because this one is probably gonna be a little better for that.
Okay.
I just need to. how do I change my screen? Okay. So here you guys are seeing the whole screen there because it's not showing up for me here.
We're seeing your PowerPoint slide
From before. The first
Yeah,
one.
not in presentation mode though.
Okay, so let me stop sharing and try again. Let me just see one thing.
You know, I could have walked you over here.
I know it's not even that, is because since I'm gonna have to leave a little earlier, I'm closer to my car.
Okay.
That way I don't have to just run. Let's see now. Where is Zoom? Okay, are you seeing the presentation now?
Yes. We're in presenter mode though.
Oh. Okay. What about now?
It's still in presenter mode. Oh, no, we're good. We're good. We're good. We're good. Don't
Okay.
touch anything.
Looks good to me.
Don't touch anything.
It's perfect.
All right, perfect. So what I had planned, and I'm gonna try to be brief, but basically just talk a little bit about the different techniques that we use in the lab, some are like that we use to answer our questions and whatnot, right? So in my lab, we're really interested in understanding what is happening to the vasculature of the brain, the blood vessels in the brain, in different neurodegenerative conditions or in normative aging, endocrine aging, etc. And we do that by making use of many different techniques that I am fairly certain very few of you have, because they're quite specific to what we do, and it may be of interest for you guys to learn something about that too. So what I'm going to focus the most today really is how we study ion channels in these blood vessels and why do they matter, right? So come on. As you probably all know, Alzheimer's disease. So this is one of the diseases that we are mostly focused in the lab. It's a neurodegenerative disease that it's fairly common in aging. The incidence starts to actually go up quite a lot after 65 years of age and up. But we still understand very little of how Alzheimer's disease actually leads to dementia, especially when it comes to the blood vessels, right? So here we have this graph just showing the incidence of Alzheimer's disease in many different areas of the country. And we can see that in Arizona, there is about 20%, 12% of the population today that is diagnosed with Alzheimer's. The problem of that, and this is probably underdiagnosed a lot because really the diagnosis mostly comes after postmortem. When we are able to look for some of those markers in the brain. But this is expected to double in the next 15 years, actually triple in the next 15 years because of the aging population, etc. The biggest problem with Alzheimer's has been that we really don't have any good drugs to treat. Like there are some new biologicals that came into the market. They don't work very well. They have a little bit of efficacy in a small subset of patients, but they have a high incidence of some potentially lethal side effects. So they're not really prescribed a lot to people. There are some other drugs that kind of act indirectly, but again, their efficacy is limited. So there has been a lot of push in the field to try to understand a little bit more of the pathology of Alzheimer's and try to find new markers. And one of these studies was here published in 2016. So what they did here is that they had this banked tissue from over 7,000 patients, and they had blood, they had cerebrospinal fluid markers, and they had a lot of MRI imaging of these patients. And they did a modeling to see how are these different changes happening according to the progression of the disease. So for example, if we look here, what we can see is that changes in neurovascular changes, basically focusing on the blood vessels in the brain, are actually happening quite early in the spectrum of Alzheimer's. So they are not only happening quite early, they are also the ones that show the most abnormalities. So the vasculature seems to be an early target for what's happening in the brain during Alzheimer's disease. But how and why does it matter? So that's really where my lab came in, and we started looking at this into a little bit more focus. But rather than focus on the neural part of the neurovasculature, we're actually focusing on the vascular part. So why are we doing that? Why does it matter, right? Because the brain is actually a very highly demanding organ. It's the most highly demanding organ in our body. It has, body weight wise, volume wise, is actually quite small, but even though it accounts for only 2% of the body weight, it receives 20% of the cardiac output. So a fifth of the blood goes to the brain at all times. And not only that, the brain has very little nutrient reserve capacity. So in the neurons, the main cell type in the brain, or at least the ones that give us cognition and whatnot, they're highly metabolically active. So whenever a neuron starts to fire action potentials, they consume a lot of energy, a lot of oxygen really fast, but they have no reserve. So imagine like this, you're exercising a lot, and then you finish exercising, you go to your fridge to get something to eat, there is no food. So you need to put an order on Grubhub and have that food delivered to you in real time. And that's exactly what happens in the brain. Whenever you have an increase in neuronal activity, you have an increase in localized blood flow to that population of neurons. And that we can see here, a lot of this perfusion is controlled by these arteries that we can see here that are called the bottlenecks of the circulation. So each artery that is going down in this direction is perfusing a column of neurons. And this is actually quite effective when everything works well. The problem becomes when the system stops working. And that, if you have even small deficits in delivery of these nutrients over time, can lead to cognitive impairment. And that's really where we are trying to understand how these things are happening. So one of the things that we know happens in Alzheimer's disease is that we have accumulation of amyloid beta plaques. So one of the biomarkers of Alzheimer's around the blood vessels in the brain. So we see here in green, these are the amyloid beta plaques, and yellow and in red are the blood vessels. So we can see a lot of yellow, right? So there is a lot of blood vessels that are just filled with this amyloid beta. And in many different models, we published in the past as well on the 5xFAD model, which is a more aggressive model of Alzheimer's disease. So using immunofluorescence, we see that they're accumulating around the blood vessels. But what does it do? And that's where we came into the study. So we use the 5xFAD model, which is a fairly aggressive model around 6 months of age. We see that these animals already have a lot of amyloid accumulation in the brain, including in the blood vessels. And we see that they have like a little bit of sexual dimorphism, which is going to come into play on the experiments that I'm going to talk to you about. So this is the model that we use. It's called the 5xFAD because it has 5 different mutations. It's the Swedish, Florida, and London mutations on the amyloid precursor protein. So it's what forms those amyloid beta plaques. And 2 on one of the enzyme complex that actually processes the amyloid to generate those plaques. So using males and females of these mice, the first question that we asked was, in these young mice that have these Alzheimer's mutations, is blood flow actually changed? So we're measuring that using this equipment, which is called a laser speckle contrast imager. So what it does, like we have here an stereotax, so we anesthetize the animal, very lightly anesthetized, and immobilize the head. And then we do a little cranial window, and then we shoot the cranial window with an infrared laser. So what this will do is that once you shoot the laser on a surface, if your surface is completely static, there is nothing moving, this is the kind of image that you will see, right? So this is what is called a speckle pattern. So basically the way that the light will interact with the tissue, if there is nothing moving, if everything is completely static, you're going to have, unless you have like a perfect sphere, you're going to have these diffraction points on the light, and we see these little blobs of red. The problem is that when we're talking about biological systems, especially in the brain, nothing is static. So actually what we see is this. So there is a lot of movement, right? A lot of things that are moving. This is a change on this speckle pattern. And what's causing the light to move that dramatically are actually the red blood cells that are moving right underneath the field of view. So every time a red blood cell goes through, it causes this blurring, this shift on the speckle pattern, and then using an algorithm, we can transform that into these perfusion maps. So what we are looking here is the surface of the brain of a mouse, of a wild type on the top and a 5XFAD on the bottom. The closer we get to red means more perfusion. The more we get close to the blue means less blood. And what we saw was that in the Alzheimer's disease model, we see lower blood flow, particularly in the frontal cortex on this mice, a very young age. So this is interesting. This was quite interesting, showing that there's something going on with the blood vessels that they are not able to, at least on a normal situation, supply as much blood as they would require as the neuron would require. But that still doesn't tell us a lot, because we're interested in how are the blood vessels responding to an increase in neuronal activation. So the next experiment that we did was actually looking at that. So we can do that in the rodents quite well. Because of the way the anatomy of the rodents, they sense their environment mostly via the whiskers, right? So the whisker barrel cortex, or the somatosensory cortex in the rodents, is actually very well developed. And we can test that if we stimulate the whiskers of the mouse at a certain frequency, we see an increase in neuronal activity if we were measuring right field potentials right there. And we also see a matching blood flow response. And that is here just an example of some of these speckle patterns. So this is actually what matters on a daily basis, right? Is how are the blood vessels able to adapt to increases in neuronal activity? So we tested that on our 5XFAD mice using that same system, and we saw that they actually, when we stimulate the whiskers with a little brush, we see that blood flow, that functional blood flow response in the wild type animals, and we see that in the Alzheimer's as well, but it's way lower than it should be. So the blood vessels are not responding properly. They're not able to dilate to bring more food to the neurons so they can survive. So over time, those neurons are just going to become hungrier and hungrier, and eventually they will end up dying. And we saw that in yet another model. So this is a different model of Alzheimer's. And we also see that on the ApoE3, ApoE4 mouse model, which is a model of late onset Alzheimer's disease. So we see this loss of these functional hyperemic responses across many different models of Alzheimer's. So it seems that indeed the neurovascular component is happening. It seems to be part of the development of the disease. But that this reduction in response that we see could be a consequence of the blood vessels not working properly. It could be that the neurons are not communicating properly with the blood vessels, could be many different things. So we actually wanted to isolate the blood vessel responses. How do we do that? We use a technique that is called pressure myography. So we are able to go into the brain and get and isolate one blood vessel from that brain. And I have here a little red rectangle on it. And we are able to cannulate that blood vessel into two glass cannulas. And here we can see one of the glass cannulas and a blood vessel in the brain. And this is a blood vessel about 50 microns in diameter. So it's about the size of an eyelash. And we would have another cannula on the other side. And what happens is, let me see if I can just, because this is supposed to be a little movie, but I don't think it's going to play. Yeah. So by doing this, we can pressurize these vessels as they would be in the body and look at their responses. And when we do that, once you pressurize the vessel at a physiological pressure, their natural response is to contract, right? So that's just your physiological. What we saw was that the vessels from the Alzheimer's disease animals had a much stronger contractile response. So they are contracting more. That actually helps explain that lower blood flow that we saw in the beginning, because the more they contract, the smaller their diameter. The smaller the diameter, less blood will go through. And when we use another way of contracting these vessels, in this case with endothelin 1, which is just a normal vasoconstrictor, we see that they are overcontractile as well. So that's when we started looking at what could be wrong, right? So quickly, just going very quickly about how this process works. In the blood vessels, we have basically two layers of cells, right? We have the inside of the blood vessels, the lining of the vessels, which are the endothelial cells. And on the outside, we have the smooth muscle, that they are circumferential to the blood vessels. The smooth muscle, they have the ability to sense changes in pressure inside the vessel. So when your vessel dilates or increase pressure or whatever, they have these mechanosensitive receptors, ion channels, that sense that stretch and allow for an influx of sodium that causes a depolarization. So the membrane potential goes more towards positive. That that on its side activates voltage-gated calcium channels. And the voltage-gated calcium channels then allow for a lot of calcium to go in and cause the vessel to contract. Once the vessel contracts and the calcium inside the cell increases, that activates a negative feedback mechanism that is called a calcium spark. So basically, we have a small release of calcium from the sarcoplasmic reticulum in close proximity to these potassium channels. So the calcium that comes out from these ryanodine receptors activate these potassium channels. They are dependent on calcium. They open and allow for a lot of potassium to go out. So what that does is that it balances charges, right? So you are getting positive charges coming in from calcium, but you're also losing positive charges. So you keep everything in balance. You hyperpolarize your membrane a little bit. So what we hypothesize is that somewhere in this pathway, there is a mismatch. Either there's too much calcium going in, or there's too little potassium going out. So we tested the first experiment. The easiest one to do was actually looking at this. We can do that by just exposing the vessels to a depolarizing stimulus that is independent of stretch. And we saw that this was not the case. The ability of the cells to contract to that depolarizing stimulus was the same. So if it's not this, then it's gotta be on the potassium going out. It's not the calcium coming in, it's the potassium going out. So we tested that, right? And we have I'm gonna skip this one, there is no point. And we tested that by looking again in those isolated vessels using pressure myography. And we can block that channel with iberiotoxin. So basically, what we're doing is we are blocking the stimulus that prevent overcontraction, right? So when you block those BK channels, you see a contraction. And that's what we're seeing here. But on the animals that have Alzheimer's disease, both the males and the females, the response to iberiotoxin is really not good. They're not contracting as much. So the channel is not working properly. And that's really where we went the next step, which was to look at the channel itself. And we do that by doing electrophysiology. So we go, again, we get those blood vessels, we isolate those blood vessels from the mouse brain. We do a mild enzymatic digestion, as we can see, and then we end up with the isolated single cells, and we can see an image of that right here. And then we can poke these cells with a little microelectrode, where we can measure currents of different channels. So we have this microelectrode, it's just a little glass. Can you guys see it there? Oh, there you go.
Yeah, I'm just gonna fix it because I think we're about to lose power. But let me confirm, is this the volume one?
The one that's connected to the wall, the volume.
That's the volume one? Okay, we're gonna lose the volume one so we can keep the screen.
All right. So we have the micropipette going into the cell, and we touch that micropipette to the membrane of the cell, and we form a very strong seal. So the seal is so strong that no ions actually get through in the border between the pipette and your cell. And we just hope that in this little patch of membrane that is usually about a micron in diameter, we have the channel that we want to study in there. And if it does, then we pull the pipette back out with that channel, and we can study the potassium ions that are going through. And we end up with these little currents over here. So every time we see this uptick, that's a BK channel that opens. And we can measure that, right? We can pretty much just count how many times the channel opens per unit of time. And we end up with this map with this data that are called open probability. So what is the probability that this channel is going to be open in that particular amount of time? And what we see is that on the wild type animals, there is a lot of channel openings, but in the 5XFAD mice, there is a lot less BK channel openings. And that was statistically significant when we looked at a different range of voltages. So in the females, the channels are not working properly. We're seeing this overcontraction just because the channels are not opening sufficiently. So the cells are more depolarized. Because they're more depolarized, they are more contractile. Right? So, but not in the males. In the males, the story is different. It seems that in the males, the amount of BK channel openings, despite being overall smaller and lower than the females in general, there was no difference between the control and the wild type mice. So that led us to start looking at this a little bit more, right? So let's go back to our little diagram, right? I told you that in order for physiological activation of BK channels, we need to have this calcium release from the endoplasmic reticulum, or that is called a calcium spark, right? If we don't have this, you don't have BK channel activity. The measurements that we were doing here are independent of that because we're taking the channel out of the cell and we're providing a lot of calcium. So this is just the channel alone. But here we're actually looking at these coupling mechanisms, right? So we can do that by, again, looking at the pressurized artery. We can load these arteries with a calcium-sensitive dye. So every time we have a local increase in calcium, and I want you guys to pay attention to these arrows, we see an increase in activity of this, this is one of the events that lead to activation of those BK channels, a calcium spark. So let me run this movie and hope you guys can see. You see these little blips, like that increase in fluorescence? So those are those intracellular calcium events that lead to activation of BK channels, right? So when we quantify this, wait, what did I do here? So when we quantify this, in the males and the females, we saw that in the males, that number of calcium sparks was lower, but it was higher in the females. So suggesting that even though we see in the whole vessel this change in the way that the BK channels are working, they are actually being caused by two different mechanisms. In the males, it's a reduction in these intracellular signaling pathways, whereas in the females, it's the channel itself that doesn't seem to be working properly. So that's what we went to investigate, and I'm just gonna show you guys a couple more things because I want to give you guys time to talk to me and ask questions. So we looked at the first experiment was like, is expression different? And we see that it's not. We did that by qPCR and Western blots. There's really no difference in expression of the BK channel. So it's not an expression problem. It's actually a problem of post-translational modifications of the BK channel. Particularly on that intracellular tail, we saw a lot of oxidation and nitro oxidation of cysteines in that intracellular domain. And what they are doing is that they are reducing the sensitivity of the channel to calcium. So you need a lot more calcium in order to activate the channel. And that's why we see that lower probability of opening that we saw on the patch clamp electrophysiology experiments. And we know that because if we add DTT or ditriethol, which is just a powerful reducing agent, so what this will do is that it will remove all of the reactive oxygen species that are on those cysteine residues. We recover BK channel activity. It doesn't go back to the levels of the wild type, but it goes about it. We have a 50% recovery, right, using patch clamp electrophysiology. And the same on the pressurized vessel. So if we add the BK channel blocker ibuprofen, we see a small response, but if we add DTT and repeat, we see that much larger contraction. So in the females, it seems that this is caused by an accumulation of these reactive oxygen species, in particular peroxynitrite, which is a nitric oxide dependent reactive oxygen species. And again, we did that by looking at those isolated smooth muscle cells. And we got this dye, which is called the fluorescein boronate. which is highly specific to peroxynitrite. So this was kindly donated to us by Dr. Rafael Radi in Uruguay and Dr. Swapnil Sonkosari at the University of Virginia. So the more peroxynitrite you have, the more fluorescence intensity you have, so the greener is your cell. And we can see here on the wild type, there is a little bit of green, not a lot, but on the 5-XFAD, there is a lot more green. So there is a lot of peroxynitrite being generated in these cells, in these smooth muscle cells of these cerebral arteries of Alzheimer's disease. And that is likely what is causing the oxidation of the channel. So I think I'll stop here for now, so you guys we can talk a little bit. But if you guys want to see some more data, I can always show more. But I want to chat a little bit.
Yeah, it was great. Thank you. I say, Dr. Perez, have you looked at parasites? I know that they're pretty new in terms of, don't laugh at me. I'm asking a question, but
Right, that's a good question, Kenneth.
looking at this, because I know that parasites might be indicative of like constriction of the vasculature in the brain. I know it's not completely confirmed yet, but have you been looking at that?
So I have not in particular. So the story with parasites is a little different, right? First of all, like there's a lot of debate of what is a parasite, like because in the brain, parasites are more like the way I see it is a continuum of different cell types that are called parasites, but you have the contractile sphincter parasites, you have the thin-stranded parasites, you have the nanotube parasites, and they're mostly in the capillaries. So there's a lot of, I know at least 2 different groups that are looking into the role of parasites in capillary constriction in Alzheimer's. But in many ways, they behave the same way as a smooth muscle cell. From the vessels that we work on, they're a little upstream from the capillaries, so the parasites really don't play much of a role, but they definitely do play a role in regulating capillary blood flow. They're going to be very important for. Have you heard about like capillary stalling?
Yeah.
Yeah, so they seem to be vital for like, or very important in capillary stalling because they constrict the capillaries, and then a white blood cell just gets stuck in there, and you completely block downstream blood flow.
Yeah, whenever you were talking about the blood flow to like neurons and the no reserve with like nutrients to the brain, I was going back to my neurodevelopment course that I took, and I was just thinking of parasites. I was like, well, what about those?
Yeah, no, there's a lot of work being done in parasites by people that do like it's a little harder to work in parasites.
Yeah.
A lot of multiphoton imaging and those kind of things. And I have some issues with multiphoton, but that's neither here nor there.
I have a question, actually. So I find ROS, reactive oxygen species, very fascinating. My name is Carolyn Coppinger. Nice to meet you, by the way. And I was wondering, and this might be in one of your future presentations or it might not, but do you know the mechanism by which I think you said peroxynitriles, is that what they're called?
Yeah.
Yeah, do you know how they're formed inside, the blood vessels?
Yes, actually. I mean, I cut it short because, I want you guys to ask questions. But so here's, since you asked that one, I'll just share.
The supplementals.
So let me pull. Can you guys see the screen again?
Yes.
We can.
Yeah. So what our data are suggesting to us is that peroxynitrite is really formed from a reaction between nitric oxide, which in blood vessels is produced a lot, right? Because that's what endothelial cells usually produce to make blood vessels dilate. They produce a lot of nitric oxide. But it's very well controlled in normal circumstances, in normal situations. But the nitric oxide, if you have superoxide in the environment, they react promptly with that nitric oxide and generate the peroxynitrite. So what we saw is that in these Alzheimer's animals, we have an increase in expression of an unregulated form of the nitric oxide synthase enzyme, so the inducible one, that is usually associated with inflammatory responses, right? They provide that nitrosative burst in macrophages to kill bacteria, to kill infected cells, etc. But in the Alzheimer's, because of that amyloid accumulation, there is a lot of local inflammation that happens. And that local inflammation is leading to an increase in expression of iNOS in the vascular wall. And that increased expression of iNOS in the condition where you already have superoxide, because we also show that, I didn't add the data here, but we show an increase in superoxide, is likely what is leading to this increase in peroxynitrite. And what the peroxynitrite is doing is actually nitrosylating the BK channel. And then by nitrosylation of the BK channel, is shifting the way the channel responds to calcium. So your normal signal, your normal calcium spark is no longer sufficient to activate the channel. So you need a lot more calcium to get over that. And if you get to that point of calcium in a normal cell, that usually tags your cell for apoptosis. So you start losing a lot of that regulation. And it happens in humans as well. So I was quite excited about that. We see that increase in nitrosylation also due to peroxynitrite in tissue from humans. So it seems to be a pattern of things that are happening in the as a consequence of the Alzheimer's disease as a whole. And just like to go one step further, we know that it's iNOS because we're generating an Alzheimer's iNOS knockout mouse. And in this case, it's not a full knockout yet, but just removal of one of the genes for iNOS, we see a recovery of BK channel activity using patch clamp and also on the isolated vessel. So it seems that, long answer to your question, yes, the peroxynitrite is likely coming from this interaction between excessive nitric oxide produced by iNOS and your superoxide that is normally high in the disease vasculature anyway.
Awesome, thank you so
Make
much.
sense?
May I ask a quick follow-up question?
Yeah.
Is your lab investigating any molecules to combat that?
Yes, we are. Yes, we are. We actually have a study. Again, I'm gonna share it. I should have just presented everything. So actually, yeah, so we are working in collaborating with a colleague, Dr. Meredith Hay, that she has this activator of a MAS receptor. So it's one of the receptors that is activated by angiotensin 1 through 7. And if you guys know that angiotensin 2 is very important in the cardiovascular system, is associated with vascular diseases, etc. But the other molecule in that system, which is the angiotensin 1 through 7, acts via a different receptor, in this case, the MAS receptor. And in the vasculature, it increases blood flow, it decreases oxidative stress, it decreases inflammation. So we actually tested this in our mice, and we used a compound that she created that is it's a glycosylated form of PNA5 of the Ang 1 through 7 that has a better brain penetration and a higher half-life. And what we are seeing is that in these conditions, if we treat our 5-XFAD mice with PNA5, we recover some of that BK activity. And it's so well recovered that if we add a reducing agent after the PNA5 treatment, so this was a treatment for 1 month, there is no added benefit. So as much as we can recover the channel, PNA5 is actually doing that. And then the next experiment we saw, again, on the blood vessels, we see that the blood vessels are constricting more to iberiotoxin, suggesting that BK channel activity is improved. And their responses to that whisker barrel stimulation are also preserved in the model. So, but so this is still very pilot, very preliminary. We're still trying, we still need to do the peroxynitrite experiments, reactive oxygen species, to see if that's the pathway that PNA5 is leading to these improvements. But we are hypothesizing that it is, based on previous data.
That's really, really cool and exciting. One of the things I was wondering about is that the downstream pathways of that really overlap a lot with what you would expect estrogen receptors to regulate. And of course, there's so much talk and has been for so many years about the role of hormones and menopause in Alzheimer's pathology. And you're talking about amyloid beta, which people who do that love to talk about. So really, my question is, to what extent is this BK channel actually regulated in its excitability by hormone receptors? And is that actually different in your animals, or is it kind of a coincidental difference there?
It's not a coincidental difference. Oh, so BK channels, right? So again, because of time, I didn't go into a lot of details. But BK channels are really formed by three different subunits. You have the BK alpha, which is all of the data that I showed today, which is the pore-forming subunit. But then you have the accessory proteins, which are the BK beta and the BK gamma. And the BK beta in particular actually has a binding motif for estrogen. So estrogen binds to the BK beta. And when estrogen binds to that subunit, it actually sensitizes the channel to calcium. So even small amounts of calcium are able to open the channel. That is one of the hypotheses why women are protected against cardiovascular disease until they go into menopause. And we actually published in 2022 that in a model of chemically induced menopause, we see a reduction in BK channel activity in the blood vessels. So it's not entirely coincidental. There is, and if you look at the numbers, so all of this, most of the stuff that I showed you guys today has been published, but if you look at the numbers on that publication on BK channel open probability between the males and the males have 20% of the total activity that you see in a female. So there is that part that females have a much higher BK dependent regulation of their blood vessels, which also brings to the other side that, once you lose that pathway, it's likely that you're going to have a much larger effect in the females than you will in the males. Whether or not that's true, we're still investigating, but yes, estrogen it's not coincidental, and it does play a role in all of this.
So the follow-up that I have for that is, and this is kind of biased towards my interests, has anyone looked at bisphenols and their ability to bind to the BK beta binding motif for estrogen?
That's a good question, and I do not know the answer to that.
Okay.
But it's definitely something worth looking at. I would be surprised if no one has. So people who like to do electrophysiology, they like to look at BK because the currents are so huge and easy to work with, for an electrophysiology experiment, right? I would be surprised if no one ever looked. I am not aware, but just because I never looked for it either. But it's certainly something that I'm actually gonna write down to look at. All right, I can probably get one more question.
I'll ask one more quick question. Earlier you had touched on how amyloid beta plaques are accumulating on blood vessels, and that's how you know how cerebral blood flow is part of Alzheimer's pathogenesis. My question is, are the A beta plaques accumulating on blood vessels and therefore affecting blood flow, or is it that the cerebral blood flow is dysfunctional and A beta is accumulating on the blood vessels? Kind of like two separate mechanisms happening at the same time, if that makes sense.
Yeah, it does. And the answer is both, actually. So there was a paper published maybe a month ago showing that you start seeing some vascular dysfunction even before you see any detectable plaques. So there is something happening in that milieu around the blood vessels that is changing the way they work, especially because the function, so these vessels, they're never static, right? So in your brain, they're always doing this, right? They are at a frequency of about 0.1 hertz. which is important for lymphatic flow. It keeps your CSF flowing, and with your CSF flowing, also clearance of these molecules. So this study, they show that actually the blood vessels start losing a lot of this rhythm before you see detectable plaques in the blood vessels or in the parenchyma. So there is something happening in the early stage of the disease that is affecting the blood vessels. Once you start affecting them, you start losing that lymphatic drainage. So more crap accumulates around the blood vessels, and then you start seeing the plaques form. Once the plaques form, it's like adding a very tight rubber band to your vessel. Like they can't do this anymore because you just added that very fibrillar, strong kind of rock-like boulder around your vessel. So it generates this feed-forward mechanism where you start accumulating more and more and more amyloid around the blood vessels.
Right.
Also, accumulation of amyloid around the blood vessels causes smooth muscle cells to die. It causes breakdown of the blood-brain barrier, leads to more neuroinflammation. It just makes everything worse. When I started my lab, actually, that was the question that I want to answer was, are the accumulation of these neurotoxins, right? These misfolded proteins in the brain, in Alzheimer's, Parkinson's, etc., a consequence of a loss of blood vessel function, or is like there's something else happening in the blood vessel that causes all of these pathways to go awry? And it seems that there are some small local pockets of neuroinflammation that are kind of leaking into the blood vessels and then changing the way they function. Because it changes the way they function, then you lose the neurovascular coupling, you lose your grub hub, and you also lose your waste management. So you no longer deliver nutrients and you don't take out the crap. So then you end up having a lot more accumulation and a lot of neurodegeneration that happens.
Right. Thank you.
Thank you.
It's interesting. You just answered the question that I was about to ask.
Yeah, it's the whole lymphatic thing. It's pretty hot right now.
Yeah. Although I do have another question, and that is this: if this kind of a issue can occur with some of these other neurodegenerative diseases like Huntington's chorea or some of the prion diseases, because there's a lot of stuff that happens there with folding of proteins, etc.
So all of this similar neurovascular dysfunction has been shown in Parkinson's, in Huntington's, in stroke, in multiple sclerosis, and the glymphatic too. Like the glymphatic, you name a disease, and the glymphatic system is not working. And at its core, the glymphatic drainage is dependent on neurovascular coupling. So you start messing with one, you mess with the other. But yes, they have been. Now, whether or not the same pathways are involved, that's still up for grabs. Like there's not a lot of people working in this field right now. I've been wanting to work in Huntington's for a while now. I just need more money and more people.
Yeah, thank you.
No, thank you. Sorry, I'm gonna have to leave, but
That's fine. Thank you.
If you guys have any more questions, Sophia has my email. Please email them to me, and I'm happy to pop up at some other time to talk more.
No worries.
Thanks.
Thank you.
Thank you so much.
All right. Thank you very
Good luck with
much.
your family.
You have a great day.
Okay.